Hi, today we're going to take a look at transport across the membrane. Specifically, we are going to take a look at active transport. So let's get started. Recall that the plasma membrane is made up of a double layer of phospholipids with protein channels and embedded proteins and carbohydrate chains. Also recall that the plasma membrane is semi-permeable. What this means is that the plasma membrane allows some substances to freely cross the plasma membrane, while other substances are not able to cross the membrane. Again, substances that are able to freely pass through the membrane are substances such as small molecules or anything that's hydrophobic. Some examples of these are oxygen, carbon dioxide, and water. There are other substances that are not able to pass through the membrane at all and are going to have to use active transport in order to either enter or leave the cell. Molecules like large molecules or molecules that are hydrophilic are not going to be able to easily pass through the membrane. So we're going to take a closer look at active transport. There are three types of active transport, just like there are three types of passive transport. There are pumps, endocytosis, and exocytosis. Let's talk about these words first of all. Pumps are pretty straightforward. They're pumping stuff into and out of the cell. In endocytosis, I'm going to highlight the prefix endo. What that means is to enter. So if we take a look at that prefix endo, en, and enter, en, also, so they have the same first two letters. This means to enter the cell. So things that are entering, so substances are going to be entering the cell. If we take a look at exocytosis, this prefix exo, it's also the same prefix in the word exit. So that ex there, things are going to be exiting the cell. So again, this should help you remember when on a test or a quiz, when you're dealing with either endocytosis or exocytosis, you know that things will be entering a cell if you're dealing with endocytosis. If you're dealing with exocytosis, things are going to be leaving the cell. All right. There are transport proteins involved in active transport, specifically with regards to pumps. There are channel proteins that are embedded in the cell membrane and end up creating a pore for materials to cross through. So if you take a look here, things are moving from an area of high concentration to an area of low concentration. These channel proteins are most oftenly associated with passive transport, which is facilitated diffusion. Okay, so it's creating a pore. These channel proteins create a pore through the membrane, and we can see here that these particles are moving from an area of high concentration to an area of low concentration. Transport proteins are also going to be involved in active transport as well, specifically carrier proteins. Now, carrier proteins actually change shape and move material from one side of the membrane to the other. The key here with carrier proteins is that energy is going to be involved, and that's the same for all types of active transport. Energy is going to be expended by the cell in order to move materials across the membrane, whether it goes into the cell or out of the cell. Material energy is going to be used in order to do that. So active transport, again, requires energy or ATP. ATP, this molecule ATP, is the molecule that cells use for energy. So active transport requires energy. It moves materials from an area of their low concentration to an area of high concentration. So again, we're moving against the concentration gradient. If you think about it, active transport is just the opposite of passive transport. Requires energy, is moving materials from a low concentration to a high concentration, and we're going against the concentration gradient. An example is a sodium-potassium pump. This creates, most often you find sodium-potassium pumps in your nerve cells. Your nerve cells are responsible for sending electrical impulses that allow you to move. Now, a sodium-potassium pump is responsible for creating the electrical gradient in your nerve cells so that your nerves can send that electrical impulse through your body and you can actually be able to move. So we're going to take a closer look at active transport. We'll move this down so we can look, and here we go.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So again, active transport, it requires energy or ATP. We're moving materials from an area of their low concentration to an area of high concentration or against the concentration gradient. So movement in vesicles, substances such as macromolecules, and remember those macromolecules are your proteins, your carbohydrates, your lipids, and your nucleic acids, and other nutrients are too large to use pumps. So we're going to use either endocytosis or exocytosis in order to bring those substances into or remove those substances from the cell. A vesicle, just think of a vesicle as a smaller version of a vacuole. A vesicle is used to move substances. It's not used for storage. It's just fleeting inside the cell, used to move substances either into the cell or out of the cell. We're going to take a closer look at endocytosis. Now, endocytosis is when molecules are moved into the cell by vesicles that form from the plasma membrane. Now, there are 2 types of endocytosis. There's pinocytosis and there is phagocytosis. In the diagram seen here, we see the cell membrane. Some molecules are entering a pocket that's being formed by the cell membrane, and the cell membrane is pinching off in order to create a tiny little chamber or a vesicle in order to transport those molecules into the cell. Pinocytosis is a specific type of endocytosis, and it's responsible for the transport of solutes or fluid. So again, here we see this extracellular fluid. We see the membrane pinching in to create a tiny vesicle, and then that vesicle is going to be moved via the cytoskeleton further into the cell in order to use that extracellular fluid for whatever function the cell needs it for. Our other type of endocytosis is phagocytosis. Now, in phagocytosis, this is the transport of large particles or whole cells. So again, here we see either a bacterium or a food particle, again, being engulfed by the plasma membrane that's being pinched off, and then we've created our vesicle that will be taken further into the cell. There's a specific type of white blood cell in your immune system called a phagocyte, and the phagocyte is responsible for breaking down any harmful either bacteria or harmful particles or possibly even viruses. So its responsibility is to go around and essentially eat up any whole cells or viruses and stuff before they can cause further damage to your body. Exocytosis is the next or last active transport. Exocytosis is when molecules are moved out of the cell by vesicles that fuse with the plasma membrane. So remember we talked about the endoplasmic reticulum and the Golgi apparatus, specifically the Golgi apparatus, as responsible for modifying packaging proteins and other things for export out of the cell. So what the Golgi apparatus does is it creates a transport vesicle, and that transport vesicle is shuttled out of the cell along the cytoskeleton to the plasma membrane, where this vesicle fuses with the plasma membrane and releases protein or other substances out of the cell. So what we can infer from this is that vesicles are made up of the same stuff that the plasma membrane is made up of, and that is our phospholipid bilayer and possibly any protein channels or proteins with their carbohydrate chains. Now, just to recap, there are 3 types of active transport. There are pumps, there is endocytosis, and there is exocytosis. Pumps are specifically used to pump things against their concentration gradient, and we talked about a specific type of pump, and that is a sodium-potassium pump, and that was using energy in order to pump either sodium and potassium against their concentration gradient, specifically seen in nerve cells in order to create an electrical gradient. In all types of active transport, energy is required. So energy is being expended by the cell in order to move substances either into or out of the cell. And with all types of active transport, molecules or materials are being moved from an area of high concentration to an area of low concentration, or up or against their concentration gradient. So hope this was helpful, and good luck.